My name is Evelin Munoz, and this summer my project is dedicated to analyzing the genetic diversity of the severely endangered Amsonia kearneyana. This species is faced with major anthropogenic and climate change-related challenges, resulting in a very small and fragmented population on a single mountainside in southern Arizona. Genetic diversity is one of the cornerstones of evolution and is what makes all the different traits we see in plants possible. This variety is what makes plants adaptable to changing environments. Moths are theorized to be the primary pollinator for kearneyana. Pollinators are responsible for a majority of the gene flow between individuals. The more gene flow there is between genetically different individuals, the more genetic diversity there is. We will be testing for gene flow between the subpopulations of kearneyana to see if pollinators are traveling between them and comparatively evaluating its genetic diversity. All of this begins with DNA extractions. First, our samples are labeled and ground into a fine powder. Then our CTAB master mix is prepared and added to each tube. After incubating at 65 degrees for an hour, chloroform is added and the mixture is transferred to a fresh tube to be centrifuged. Afterward, we carefully isolate our supernatant, add alcohol, and allow samples to precipitate in the freezer for at least an hour. Then samples are once again put in the centrifuge to pellet our DNA and go through a series of drying and washing steps. After the final drying step, the DNA is resuspended in DNA-free water. Once all our samples were extracted, we created our DNA library by digesting, barcoding, and size selecting our sample before we ran a PCR. Once amplified, we cleaned up our sample with magnetic beads. And with that, our library was ready to be sequenced and analyzed to give us our results.